Standard aqueous biocatalysis relies on random collisions in dilute environments. In a traditional bulk reactor, molecules are constantly diffusing, making interactions a matter of chance. Because these bulk cultures lack tight physical confinement, fragile intermediate molecules often degrade before they can react with their intended targets. This thermal and structural chaos makes it highly inefficient to synthesize rare or complex secondary metabolites. We can solve this geometric problem by repurposing natural biological architectures. This is the pollen microbiome microbioreactor, or PMMB. Rather than treating pollen as a simple allergen or a passive delivery shell, we engineer it into an active, biased microenvironment. By strictly controlling the physical shape and scale of the container, we change the thermodynamic and kinetic outputs of the reactions occurring inside it. To harness this natural architecture, the raw sporopollenin must first be completely stripped of the proteins that trigger human allergies. One processing route uses acid extraction. This dissolves the organic core, leaving behind robust, empty shells called sporopollenin exine capsules, or SECs. An alternative route uses alkaline softening, which hydrates the shell and transforms the matrix into a castable microgel. Regardless of the extraction method, both processes preserve the critical structural asset, the highly porous, cross-linked aliphatic aromatic walls. Before any microbial testing begins, every batch must clear a strict quality assurance gate. Lot-level IgE ELISA tests must read completely below the threshold of detection to ensure the material is de-allergenized. What remains is a chemically resilient engineered framework, safe and ready to host specialized microbial catalysts. Inside the matrix, tubular channels measure exactly 200 to 300 nanometers across. Fluid moving through these spaces establishes localized microgradients, shifting pH continuously. The walls are heavily embedded with hexagonal aromatic rings, creating dense electrostatic forces that actively trap and align aromatic substrates parallel to the wall. By constantly pulling substrates out of the fluid flow, the matrix forces extreme physical crowding right at the channel surface. When we introduce isolated enzymes or live microbes into this compressed layer, they are forced to process these perfectly aligned molecules over other suspended options. The combination of spatial crowding and aromatic adsorption functions as a precise thermodynamic lever, actively steering metabolic pathways away from the random outputs expected in a bulk culture. Configuring these microenvironments involves too many variables for physical trial and error. The physical chemistry must be modeled computationally. We use molecular dynamics docking to test how different molecules fit within the matrix. This generates a ranked list of binding energies, measuring exactly how tightly specific substrate shapes will lock against different pore wall chemotypes. Next, we apply constraint-based modeling, specifically a technique called FBA Light. This algorithm maps the exact metabolic flux of GRAS-certified microbial strains, pinpointing where pathway bottlenecks will occur as substrates pass through the nanochannels. The mathematical outputs from these bottleneck predictions dictate our exact design of experiments parameters, locking in the required pH and co-substrate ratios before a single wet lab run. We calculate the exact thermodynamic requirements in silico, ensuring we force the targeted reactions rather than hoping to stumble upon them. For human nutraceutical applications, our primary chemical target is routing L-tryptophan pathways to synthesize novel indole-phenol hybrids. A secondary objective uses these same spatial constraints to drive the enhanced L-glycosylation of flavonoids, a specific structural modification that radically improves their solubility and shelf stability. This graph charts the mass spectrometry outputs. To prove the reactor is actively biasing the chemistry, the PMMB system must output at least a 20% increase in unique molecular features compared to the bulk culture control line. Synthesizing new chemical structures is only the first requirement. We then test the safety and efficacy of these new metabolites using human organ-on-a-chip models. The new metabolites must bind to and activate the AHR receptor, a key immune regulator, but they must fall strictly within a specific tolerance window to prevent cellular overstimulation. Simultaneously, we monitor the physical integrity of the tissue barrier. Readings for the tight junctions between the intestinal cells cannot fluctuate by more than 10%. Clearing these precise biological checkpoints confirms that the reactor yields potent immunomodulators without degrading delicate tissue membranes. Because the mechanism driving these reactions is purely biophysical, the reactor framework is completely substrate agnostic and freely adaptable to different biological domains. We can lift this exact physical model from human tissue applications and deploy it directly into the soil at the plant-root interface known as the rhizosphere. In commercial forestry, we deploy this by encasing seeds in SEC microcapsules, or by planting root-adjacent microgels loaded with plant growth-promoting microbes like trichoderma and bacillus. Once in the soil, the matrix releases concentrated phenolics to prime the plant's immune defenses, while the microbes output localized indoles to actively shape the physical architecture of the growing root system. Empirical field data tracks the impact of this local thermodynamic steering. Treated saplings yield a 25% increase in root-to-shoot mass and a 30% reduction in disease incidence. The precise biophysical constraints used to manufacture human immunomodulators are directly applied to fix systemic establishment failures in commercial timber. Beyond static deployments, the structure operates as a modular hardware platform, fully capable of integrating external mechanical upgrades. For example, the directional abscission gating module adds an asymmetric anchor to the pollen shell. It physically restricts the reactor from detaching unless it is struck by a favorable wind flow orientation. The system is equally adaptable with input chemistry, accepting complex feedstocks like cannabis leaf phenolic fractions to drive the targeted synthesis of novel flammable alkaloids. From initial computational modeling to leveraging nanometer-scale geometry, this framework executes targeted biocatalysis across multiple kingdoms of life. By utilizing the material intelligence evolved within Sporopollenin, we establish a rigid baseline for bioengineering. The physical container is now as active and crucial as the catalyst placed inside it.